Okay, hello remote practical fans. So this is obviously a bit of an unusual way of doing a practical, but this is what we're going to do. So you can still achieve a load of those required practical CPAC standards even while you're not in school. So required practical 4 is one we've been putting on for a while. It is on membranes and membrane permeability. So it links to the topic about getting in and out of cells. So it links to phospholipids, getting in and out of cells, diffusion, osmosis, membrane permeability, and indeed the bit of the syllabus about water. Okay, about the polar nature of water. So we're going to crack on then with a little bit of a reminder of the theory. Now, if I show you this, you should recognise it as the fluid mosaic model. Okay, you should recognise all of those bits that are labelled. Okay, if not, that is a glycoprotein sticking off protein there. That's a glycolipid. That's a carbohydrate chain. That's a phospholipid. That's some cholesterol in there. That's a channel protein, and so on and so forth. And if you don't remember all of those things, then go back to your notes on that topic and do a bit of revision before we carry on, because it's this bit up here, the nature of the phospholipid, that we're particularly interested in for this practical. So the idea of why membranes stick together in this phospholipid bilayer, where the tails stick to each other and the heads stick out, is all to do with charge. Okay, water is a polar molecule. Now, you may have done the water lesson and know all about that, and it certainly would have come up in the hydrogen bonding and all those other areas of the syllabus where water pops up. Okay, now ethanol, the liquid ethanol, our pure alcohol, is a non-polar molecule. Okay, so water is a polar environment. Ethanol is a non-polar environment. Now, you should remember, look, that phospholipids have got this hydrophilic phosphate head, which is polar. So that will dissolve quite happily in water. Okay. They've also got hydrophobic fatty acid lipid tails, which will not dissolve in water. They would much prefer to dissolve in an uncharged environment like ethanol. Okay. So the phosphate end is obviously very soluble in water. The hydrophobic fatty acid tails orient themselves away from water or will more happily dissolve in non-polar environments. Okay, so the theory of this practical goes as follows. We can mess with the membrane if we change the polarity of the environment, and we do that by adding more ethanol. Okay, so the theory goes like this. The higher the ethanol mixed in with your water, the less polar the environment is. Okay, the less polar the environment is the less able those phospholipids are, that bilayer is, to hold together. Okay, and the higher the ethanol concentration, the lower the polarity of the environment, the more leaky the membranes get, because these phospholipids start to sort of not be held together by those two polar environments, those watery environments on the outside and the inside of the cell. Okay, that's the idea, that's the theory behind this. Okay, so how can we actually visualize that effect? Right, so we've got the graph here. Look, as you can see, ethanol concentration increasing up there and the permeability of the membrane increasing like so. Right, the question is, how can we actually see that? How can we visually see it? Well, if we were to use just any old cell, it might be that the cytoplasms that might be leaking out, the contents of the cells that might be leaking out, may have no colour to them whatsoever and make them very difficult to see. So for this practical, we're picking a type of tissue that is plentifully available and has a very, very strong dye in it. And we all know that beetroot has got a very, very strong dye within the cells. Okay, so this is called betalain. Obviously, you can pause this video and have a read about the chemistry of the beta-cyanin family of pigments, which is what beetroot has. Okay, betalain is the sort of generic term for what we're going to make leak out of these cells. Okay, so, and obviously then you've all got the fun there of the effect of betalain on the digestive system and so on and so forth. Okay, right, so that's good. So we can see the betalain. So we're going to increase the amount of ethanol in our mixes. Like that will allow more and more of this betalain to leak out. Okay, that's visual, right? But in science we like quantitative answers. We like answers we can put numbers to. And redness or pinkness, right? Pinks per minute or whatever, is not a very good unit, right? So how can we actually turn that into a measurement? Well, you should be familiar with this bit of equipment called a colorimeter. Okay, now that's obviously one form of colorimeter. I quite like those colorimeters, they're very nice. You might have used the ones that attach to our multimeter thingies, so you've got loads of different meters you can plug in and just have a colorimeter attachment. That's all perfectly normal, but they're all doing the same thing. They're shining light through a sample and then measuring the percentage of light that gets to the other side. Okay, and obviously they'll give you a result in either transmission, what percentage of light made it through, or absorbance, which is more usual, what percentage of light didn't, how much light did the pigment absorb. And obviously the more light that makes it through to the other side, the less pigment there are. Now with this one, because it's red colour, we use blue light, okay, usually because blue light gets very effectively absorbed by red things. Go back and consult your GCSE physics if you're not sure why that is. Okay, so colorimetry is how we're going to measure it. So in terms of the variables, what you're changing, what you're measuring, you should know in your sleep now that independent variables are things I change. Independent I change. We're changing the percentage of ethanol compared to water in each of the little experimental tubes. So you might have pure water at one end, pure ethanol at the other, and then some percentages, dilutions in between. Okay? And one of the skills we're assessing here is your ability to design dilutions. Now, the dependent variable obviously is then absorbance. So once you've done this, you can take the liquid off, put it in a colorimeter, and you measure how much of the betylene was freed from the cell by that percentage of ethanol. Right? Now I'm going to leave it up to you to come up with, well, how are we going to make this a fair test? What are we going to actually control in terms of our control variables? Okay? And indeed, what would be a suitable control experiment for this? Okay, that's all stuff you should be very familiar with and be able to come up with. So, in short, the method looks like this. You cut your beetroot up into chunks, you mix the different concentrations of ethanol solution together, you place the beetroot into the solutions and you wait for the membrane disruption to occur. Okay, you drain off the solution, you keep that solution, okay, drain it off into a cuvette, you measure the absorbance of the solution in the colorimeter, okay, and that gives you your results. Now obviously it's a very, very sort of surface level method, right? I want you to be thinking about what the controls are, what the health and safety is, you know, all the normal practical skills that we're trying to get across to you through these required practicals so as to make you a better scientist. Okay, these are standard skills. So that's the method in short. Right? Hopefully you all understand the theory, how it works now, and in overall sense what we're intending to do with this practical and why it's here. Okay. So the tasks you're going to be looking at and you're going to be assessed on for this one. So I want you to print the practical method ready to stick into any lab books. Okay, that might be quite a way down the road, but there we go. Complete the table to show which volumes to add to produce the colour standards. Make a written risk assessment, stick in your lab book. That is a required and often undervalued and lack of attention skill. And design a table to use during the practical to record your results. So if you were doing this practical, how would you design a table for that? Okay, because that is a required practical skill as well. And obviously you should know how to draw a table now, but please refer to the conventions and the student practical guide if you are unsure. Okay, now if I can just have a last little word about risk assessing. This is usually done, but it's usually poor by GCSE and A-level students because you've been spoon-fed risk, right? You've been spoon-fed risk management since year 7 because we don't want you losing a finger or an eye in year 7. Okay. Now, obviously, that means that you come out intact of GCSE science, but it makes you very, very bad at actually doing risk assessments. What a good risk assessment should do is assess and manage the risks of the practical. Okay. What it doesn't do is manage the risks of you, right? So don't say you're rubbish, right? Oh yeah, I need to be doing this 5 miles away with big tongs in a bunker because I might drop the beetroot on my toe or something like that. Yeah, you're A-level students. You know, I'm assuming you're not going to stick a test tube in your eye. I'm assuming you're not going to bludgeon your partner to death with a beetroot, right? All these things are not good risk assessments because, I mean, yeah, maybe if you got a ton of beetroots dropped on your head, then it might hurt a bit. But is that a relevant risk to this experiment? Not really, right? It's not the same, okay? You're also not trying to eliminate every bit of risk. You're chopping up a beetroot and sticking it into methanol, right? So I need to wear a full hazmat suit and big gloves and stuff because I'm using a little shotgun. No, this isn't only as dangerous as cooking at home, right? It's about managing risk. Okay, it's about the idea of what you do reasonably to adjust for things that actually are risks. Okay, you must not also say that all risks are the same. There's obviously this tendency to come up with as many risks as you possibly can because that's a better risk assessment. No, I mean the risk of a meteor hitting the lab while you're working in there is not the same as the risk of you breaking a test tube. Okay, the risk of breaking a test tube is much higher. The consequences of breaking a test tube aren't really very significant. You just clean it up and move on, right? So they're not all risks are the same, and you've got to actually put them in some kind of order. This is the primary risk, and here's what I'm going to do to control it. And you are way beyond now as A-level students putting the burden of safety on other people, writing things like, do what the teacher says, listen to instructions at all times. No, you are in charge of your own health and safety, and this is the skill that we are assessing. So make it meaningful, make it an assessment of the practical. Realistically, what about this practical might do you harm, and how are we going to manage it? Okay, and have a look at the HAS cards for things like ethanol, right, in this practical, and use that to suggest some sensible mitigations of this. Okay, so that's the practical, that's what you're doing. Hopefully you now understand a little bit about the theory and what it is you've got to do before this practical. Okay, there will obviously be assessing sets of data coming up afterwards in part 2. Thanks very much.